Good evening, everyone. Can everybody hear me okay? Welcome to the 2017-18 series of Let's Talk About Health. It's really nice to see everybody here, and it's nice to see so many familiar faces. A few housekeeping points just to begin with. First of all, please can you make sure your mobiles are either switched off or switched to silent. Secondly, we're not expecting a fire alarm, so if there is a fire alarm, it means we have to evacuate. And the fire exits are signed, and the student helpers who are around will tell you exactly where to go, but basically we leave the building. So my name's Karen Chapman, and I work in this building. So you're in the Queen's Medical Research Institute, which is one of several university research institutes on this site now. And you'll see from all the stuff going on that there's more being built around the campus. So we have two speakers tonight, as usual. We have a clinician and a scientist. We have Richard Mellenby, who's currently the head of companion animal science at the vet school. And we have Iris Mayer, who's an immunologist based out of the vet school. Some of you may recognize Iris. She has been one of the hugely valuable volunteer helpers that we have, and she's led a number of lab tours for the school pupils in previous years. So it's really nice to have you on the other side of the fence, Iris, and hear about the research that you're doing. So without any further ado, I'd like to welcome Richard to give the talk on vitamin D, hope or hype.
Well, thank you very much, and thank you for such a generous introduction and an opportunity to come and speak. And it's wonderful to see so many people here to hear about our work. So thank you very much for giving up your evening and coming along to hear about our activities. So, as Karen mentioned, I have the great honour of leading the Small Animal Hospital, which is based up at the Easterbush site, which is one of the main campuses within the University of Edinburgh. We're a very busy hospital. We're one of Europe's largest. We see about 20,000 patients. And one of the other great advantages of the work that I have here is not only do I get a chance to see animals and treat patients on a regular basis, but I'm also able to work right opposite that, really, in the Roslin Institute, which is one of Europe's largest animal biosciences centres. And because of the nature of the university, and one of the great joys of it, is that the university very much encourages people to work across disciplines. And so I'm just one of many vets that work within the medical areas of the university, where we have opportunities to collaborate not only with our medical colleagues, but people in the basic scientists as well, such as chemistry and physics. And it's a really wonderful mix where all these people come together to try and answer some of the really important questions of our time. And my lab is particularly interested in trying to understand the relationship between nutrition, inflammation and health outcomes. And a particularly important theme runs through all of our work is to try and understand more about the biology of vitamin D. And the approach that we take is perhaps slightly different from some of the talks that you may have heard in the past, because what actually stimulates an awful lot of our work is our patients. So our patients that we see up at the Easterbush site, the animals that we see have very similar and sometimes very different conditions than what humans have. But nonetheless, there's a huge number of types of vitamin D disorders that animals see. And it's these cases that we see through our clinic that really stimulate us to try and understand more about the biology of vitamin D and to really try and develop better diagnostics and better therapies to try and help these animals. And we hope that this sort of philosophy then reaches out and helps people and medics in their own activities. One of the great advantages of being a vet within this sort of research space is that we have access to some really remarkable populations of animals that have been really incredibly well studied for a long period of time that really perfectly align with our interest in nutrition. And we're going to talk about some of the stories and some of the work that we've done in some of these populations, not least in the one on the bottom left there that you can see, the soya sheep population that's been incredibly well studied by researchers at Basic King's Buildings for over 3 decades, and it's been hugely informative from lots of different points of view. And then finally, towards the end of the talk, Iris is going to talk about some of the studies that we've done within the laboratory to really start to understand how vitamin D influences the immune system. And what we hope to collectively do with this programme of work is to really try and inform this debate. This is one of the biggest public health debates I think we have in Scotland at the moment, trying to understand more about the role that vitamin D plays in maintaining health. We know it plays an important part, and we have known this for a long time, within bones, but there's still an awful lot for us to learn about how it affects other, particularly non-skeletal disorders and physiology. So just to give the talk some context and structure, there's going to be three main questions that we're going to talk you through. And really, what I'm going to try and do is to tell you a little bit about what is vitamin D. So vitamin D is a very important hormone. It's something that we can obtain and take in from both our food, but it's also quite remarkable in the fact that we can make it in our skin. So not all mammals can do this, and cats and dogs aren't great at this, but most mammals are very good at being able to produce it in the skin, and that's a crucial way upon which animals make sure that they have enough vitamin D. We're also going to talk a little bit about, well, what does actually vitamin D do? We know that vitamin D plays a very important role in making sure our bones are healthy. But one of the things that's changed, particularly over the last 3 to 4 decades, has been this explosion of interest in trying to understand what vitamin D actually does beyond the skeleton. And that's of real relevance to this institute, but to this area as well, because of the way we're interested in understanding more about how vitamin D modulates the immune system and perhaps alters our risk for multiple sclerosis, which is something that we're going to talk a bit more about towards the end of the talk. And then finally, we're really just at the very start of really trying to understand the really amazing ways in which vitamin D can modulate the immune system. And we've been using some really quite contemporary techniques that Iris is going to talk through that really have allowed us to understand a lot more about how vitamin D metabolites are able to affect the immune system and really play what we consider now really a very important mechanism in regulating the immune response. So, just very briefly about the history of vitamin D, and you'll just have to just indulge me for a moment as I introduce the most famous Melonbee of them all. So, the Melonbee is a fairly small clan, there's only about 120 of us, and almost everyone comes from Hartlepool, the Riviera of the North, as we like to know of it for those that have never been to France. Sir Edward had an incredibly distinguished career. And what he actually decided to really study and really trying to understand and do something about was rickets. So rickets was a huge public health issue within his lifetime. So particularly about 150 years ago, this was a really, really major public health problem. And particularly in this part of the world, in the industrial cities of the north, in Glasgow and Edinburgh, rickets was estimated to have a prevalence of around about 50 to 60% of all children. These were people that had poor diets, poor environmental conditions, there's an awful lot of smog. And this resulted, these sort of toxic environmental and dietary risk factors resulted in this very, very high prevalence of rickets. And he was invited by the then head of the MRC, Sir Walter Fletcher, to actually start to do some work on this. And he was offered this donation of £250 to start it. And at this stage, he was a very early career scientist. He was only about 30 when this offer came through. And as everyone in Edinburgh knows, when the head of the MRC asks you to do something, you tend to jump to it. And that's exactly what Sir Edward did. He went on and really did an awful lot of work to try and understand more about what was underlying this really major public health disorder. And so a few years later, he published what I would argue, and I appreciate I'm quite partisan in this, but nonetheless I would argue that this is one of the most important papers from a public health disorder point of view that was published in the last century. And this was a series of lectures that he gave to the esteemed College of Surgeons. And through a series of lectures, he really delineated and described some of the experiments that he did that really allowed us to really take this really huge step forward in understanding how we can do something to prevent rickets. And I'll just talk through this, because this is particularly for the school children and the sort of higher students that are with us who we're particularly welcome to see. And it's great that school children are able to come to these sort of lectures and hear a little bit more about our work. But this is a very, very nice example in this table that he included in that paper about the experimental process in itself. Because what he did, and I'll just draw your attention to these 3 columns on the right-hand side, this is diet 4, so this D4, that means just diet 4. And so all these dogs in these various groups were just fed this very standard diet, and in this case it was pretty much just bread and milk with a few other additions, but really bread and milk was the background of this. And in the 186 experiment, they added linseed oil. In this one here, sorry about this pointer. And this one here, we added olive oil, or I said we'd added olive oil, and then in the final one, it was cod liver oil. And what you can see from the text here, where we had just linseed oil and the milk and the bread, these animals went on to get rickets, and this absolute classic phenotype or appearance of these dogs with these bored legs indicated that these animals went on to get rickets. Whereas when they were given cod liver oil, these dogs were normal. So again, from a picture of the time of the experiments, you can see an absolutely normal-legged and skeletally normal dog. So this very, very simple experiment was really a huge advance in the field. And as many school children in the years that followed from this would testify, the administration of cod liver oil led to really the resolvement of this as a public health disorder within the lifetime of Sedgwood. And really very few scientists, and you'll hear lots of people in these talks come forward and talk about their work and their disease of interest, very few will actually see it pretty much eliminated. And it still is present, it's still present in the UK, but to a much lower extent than what it was 100, 150 years ago. And that really all comes back from that very simple experiment that Sedgwood did all those years ago. So in the intervening years, and I'm just going to very briefly talk through this slide, a number of key points were made and other additional discoveries were made about how vitamin D actually affects physiology. And the key things to try and take from this is that we now know that vitamin D comes from two sources. It can come from the skin and it can come from the diet. And we know that cod liver oil is a particularly good source of vitamin D, but many other sources of vitamin D can come from that, from oily fish and from certain eggs. Liver can be good sources of vitamin D as well. And in the skin, particularly in summertime, perhaps not so much in this part of the world, but certainly a bit further south, we can get very, very good levels of UVB radiation, and we can produce vitamin D as humans very readily in our skin. Now, we've tried to keep this talk as jargon-free as possible, but it is important to just very briefly understand that vitamin D itself isn't a desperately active hormone. It has to undergo two metabolisms. So it has to go undergo these two conversions, and it's this first conversion that happens in the liver that goes to 25-hydroxyvitamin D. And this 25-OHD metabolite that you'll see in future slides, this is what, if you went to your GP and they said, you asked to see whether you were vitamin D replete, that's the hormone or the metabolite that they would measure to see whether that was the case. However, what we also know is that it's this hormone here that's a second metabolite from the 25-hydroxyvitamin D is then metabolized in the kidney to this second metabolite, and this is the 125-dehydroxyvitamin D, and this is the one that's most metabolically active, and this is the one that we study in the laboratory, as Iris is going to talk through. And this is the hormone that really has this really profound effect at making sure that we have enough calcium in our bodies to make sure our bones remain healthy. And it does this through a whole series of very clever mechanisms, particularly affecting the amount of calcium that we absorb through our diet and through our intestines. So again, vitamin D is made from the skin, but it's also consumed in the diet. It then goes to the liver, where it's converted into this 25-hydroxyvitamin D, and it then goes to the kidney, where it's converted into this very active metabolite, this 125-dehydroxyvitamin D. Now, I would like to say that, I was just carrying on the family tradition, and this is what all Mellenbys do, but actually nothing's further from the truth, and I knew nothing about the work of Sedgwood, really, until I was really challenged by some of the cases that came in through our hospital. And my interest in this really stems back to Digby. This is a very famous now German Shepherd that I saw about 15 years ago with colleagues when I was training in Cambridge. And Digby came in and had very, very high levels of calcium in his blood. And we know that dogs that have very high levels of calcium in the blood, something very serious is wrong here. And this is a very serious problem, and if it's not corrected, all this calcium just gets deposited all around the body, but particularly in the kidneys and sometimes in the nervous system, and it causes huge, huge health problems. So if a dog has a calcium that's above the reference range that we can see here, if it's above the reference range and at this sort of level is 3.5 millimolars, we know that this will be catastrophic in the long term for the dog. So we really have to understand, well, why is this happening and what can we do to help the dog? And in this case, in Digby, and we had a whole series of dogs that came in our clinic at this time, the usual assays that we did to try and understand, well, what is the disease process here that's driving this problem, they all were really very, very unhelpful. And this was really Digby was the case that really kick-started us to think about ways upon which we can start to understand more about vitamin D biology within the animal themselves. And so it was this case that then encouraged us to work with collaborators in Manchester to really develop and validate an assay that allowed us to measure these vitamin D metabolites. And to kind of cut a long story short, and it was a very long story before we worked out what had happened here, is that actually the dog food manufacturing plant that made the food, the vitamin D weighing scales quite literally had broken. And so really through some very clever detective work through one of the owners, and full credit has to go to this owner for really unpicking all of this, but she was able to find a place that measured vitamin D within the food, and it was 96 times above what it should be. So he had this huge, huge excess amount of vitamin D, and that was what was making Digby very, very unwell. And so this was really the first case that really allowed us to really measure in our institute, to measure vitamin D metabolites within the patient, and really to show that this was a very, very powerful approach to try and understand these important, albeit quite rare, events. And I'm pleased to report that with supportive care and a change of diet, Digby went on to make and the other dogs we treated went on to make a very, very good recovery. And so this made us very excited that we then had an increased toolbox to allow us to really understand more about the disorders that we were seeing within our clinics. And one of the other really striking cases that we had was this dog called Poppy, who again had this incredibly high calcium concentration that was way above reference range. And this makes us very alarmed as clinicians, because we know if this goes on unchecked, it's going to be really devastating for the patient. And what we also noticed with Poppy was that her lymph nodes were enlarged. They weren't as enlarged as what we see in patients that have a malignant disorder of their lymph nodes, but nonetheless they were very, very enlarged. And we actually took one out, and this is the picture of it. And what we use when we look at lymph nodes is, when we look under the microscope, we use special stains to allow us to identify certain types of cells. And we were able to do this on several different types of stains and really understand what had gone wrong in this case. And what Poppy had was really a very specific type of inflammation within her lymph nodes, where the immune cells had gone crazy and they were actually producing too much vitamin D themselves. Now, this is a very, very unusual scenario that we see very occasionally. We can sometimes see this in humans as well, with different types of what we call granulomatous lymphadenitis. And in Poppy's case, we were able to treat with fairly straightforward treatment, and she went on and did extremely well and made a full recovery. But these two cases really show, one, how useful it is to see these cases and develop the toolbox so we can understand them. But it also does highlight the perils of too much vitamin D. And I think as we continue with our story, that's one of the things that is very, very challenging. We know that low levels of vitamin D are very, very damaging, particularly for bones, but potentially for other disorders. But we also know that too much vitamin D is equally bad. And getting that balance right is what really this whole field is trying to grapple with and understand more about. So after we developed these tests within our clinic population, we wanted to then start to use them to really understand a little bit more about how vitamin D was influencing health in these animals beyond the bone. And we knew from the work of Sedgwick that vitamin D was really crucially important for bone health. But we were really very aware that there was lots of other work ongoing that was really implicating vitamin D in the health of people beyond the skeleton. And the reason people were interested in this can all be taken back to this one paper in Science. So Science is a very, very important journal. We publish a lot of, well, I say the wrong way, it's never something I've done, but distinguished scientists will then publish their work in Science when it's of the very, very highest order. And this story that was published, 35 years ago, was hugely important because it showed that the vitamin D receptor was present on tissues that we really hadn't previously expected to be the case. And these were the immune system itself within the humans. And the receptor is very important because this is how the vitamin D essentially talks and communicates to the cell. It will interact with the receptor, and then from that, the cell gets instructions about how it should then develop and how it should grow and how it should then behave. And this is really mirrored with our own population. So this is just a slide from some dog intestines. So this is just from a dog's intestines that's then put under the microscope and stained with an antibody that allows us to detect the vitamin D receptor. And where we detect it, it stains in this particular protocol brown. So what we can see in many different areas, and this is the inside of the intestines, and this is the villus going right up to the top. So this is what we would call the lumen of the intestines, and this is within the space between the villi. And what we can see here, particularly at the outer edges of the intestine, there's lots of this brown staining, and this is a really rich area where there's a huge amount of vitamin D receptors being present. And we've looked in many different types of tissue, and we found the same pattern. Huge numbers of different types of cells can express the vitamin D receptor, and so then have this potential to have their behavior modulated by the vitamin D that then circulates around our body. Now, the reason why this was really important and why this was something that became of real interest to people was because we know that diseases don't really uniformly present across the world. Now, when we look in some of the developing equatorial countries compared to the sort of more developed countries within, say, Western Europe, there are an awful lot of sort of socio-demographic risk factors that might influence why some diseases are prevalent in one society and not in the others. But this is a particularly powerful plot that's been taken that really shows about the distribution of multiple sclerosis within a single country. So an awful lot of those sort of socio-demographic factors are controlled, and I absolutely appreciate not all are controlled, but it's an awful lot better than when we look across it on a continental scale where there really is vast differences. And what we can see here in North America, right at the top here, in Washington and Minnesota, in these dark colours here, this shows really an intense level of or high level of multiple sclerosis incidence. Whereas in the more sunnier states down at the bottom, we have this much lighter colour here on this case control study here that shows the incidence of multiple sclerosis is much, much lower. So this really chimed with these people at the same time that we're discovering that the immune cell had the presence of the vitamin D receptor. And so all of a sudden, people started to join the pieces together and say, well, actually, if the immune system can be influenced by vitamin D, then potentially this might be the reason why there is such a sort of latitudinal risk to the development and presence of multiple sclerosis. Now, this is where the great challenge comes in, and it really refers back to the title of the talk. As we'll show with what Iris is going to talk about, we've made great progress in really understanding more about how the vitamin D modulates the immune system, but it still remains a deeply controversial area as to how much of a risk factor vitamin D is, and in particular, how much benefit vitamin D is at either ameliorating your risks and particularly controversially, whether it's any benefit at all if you actually have the disease. So these observations then led to this really explosion of work. So since that study in Science 35 years ago, there's then been this huge amount of work that's been done that's really started to understand much more about how vitamin D then influences a whole range of cells, particularly the immune system, but also other areas. And on here we've got the reproductive tract, the bowel, the lung, the skin, can all be in some way influenced by the presence of vitamin D to a greater or lesser extent. And there's a huge amount of interest in how vitamin D is actually converted from its essentially precursor form, this 25-hydroxyvitamin D, into this active form within the site of inflammation. And that's a theme that we're really, really interested in our lab, about how vitamin D acts locally at sites of inflammation to really alter the end outcome of that inflammatory response. So I'm just going to talk about two particular areas of interest to us very briefly. And one is just the relationship between vitamin D and inflammation, and the other one is the relationship between vitamin D and mortality. So this is a very typical plot that we see when we try and just map out the difference and the relationship between vitamin D status, which we always assess by this molecule here, this first-pass metabolite that we talked about before at the start, this 25-hydroxyvitamin D. This is how we measure vitamin D status, and a marker of inflammation. And in this case, we've just taken a subset of immune cells that we call neutrophils. And what we see is this negative relationship. So this straight line that goes down here, there's a lot of variation from that line, but nonetheless, it always heads in the same direction. And this is a really striking observation that we've found from any of what we would call the innate immune system, the specific subset of the immune system, whether it's plasma cytokines, the things that influence the activities of the immune system. All of these markers of inflammation, we tend to see this very clear relationship. If you've got lots of vitamin D, you tend to have low inflammatory markers. If you have if you have very low levels of vitamin D, you tend to have high inflammation, whereas if you have lots of vitamin D, it tends to be much lower. And the second piece of work that I was just going to show is that the relationship between mortality and vitamin D status. And much of this stems from the work that, again, we see on our clinics. And in this case, this is just a cohort of dogs that had a disease very similar to inflammatory bowel disease in humans. It's not quite the same, but has an awful lot of similarities to the disease you see in humans. And we take the same approach that we do in human medicines, and this is just some colleagues doing a colonoscopy on a boxer dog to try and take tissues and photographs from inside the bowel to see exactly what was happening and what type of inflammation was ongoing within the intestines. And when we take our populations of dogs and we split them into two groups, ones that survived versus ones that died, what we see is the ones that survived have a much higher level of vitamin D than the ones that died. So it's important to realise that we're not saying this is causation, and this is again a very important point to articulate to the school children. This is not absolutely saying that this is the cause of the mortality, but nonetheless the relationship is very, very striking, and it's been consistent across all populations that we've studied. And so some of the things that we've done where we've just done exactly the same, we've really taken the approach of just measuring vitamin D in all the patients that come in through the hospital, so taking a very unbiased selection. And this is what we've done within our cat community that comes into our hospital. Every cat that comes in, we just measure vitamin D and vitamin D status, and we then just track these cats and see how they did and how they progressed. And this just shows the range of the type of cats that we see. This was a really lovely, fantastic cat that had a really rare disease called polyradiculoneuritis. And this is where the nerves on his limbs just didn't work. And so he was just in this very, very unfortunate and sad and upsetting position for him where he just couldn't move. And with lots of nurse and support and a bit of patience and time, he went on to make a full recovery. This was again a very sad case of a cat that went acutely blind because of an eye infection. Called toxoplasma that went on to cause, and this is why he has this sort of rather startled look. So we measured vitamin D in a whole series of cats that presented with a real myriad of problems. And what we found, and this is a sort of summary plot of our results, is that when we split the cats into 3 groups, ones that had low vitamin D status, ones that have intermediate levels of vitamin D, and ones that have high levels of vitamin D, there's a very, very striking relationship between having low levels of vitamin D and unfortunately having a poor health outcome. So it's a very similar finding as what we had with the dogs that had the intestinal disease. There's something about being in this low group of vitamin D that really does seem to associate strongly with a poor clinical outcome. So again, we're not saying it's the reason of the poor clinical outcome, but nonetheless that relationship is very, very strong. And so just at the end of my little bit before I hand over to Iris, I was just going to spend 5 minutes talking through some work that we've done with colleagues at King's Buildings, particularly led by Dan Nussey and Josephine Pemberton and Catherine Watts, that's really been part of a big team that over 3 decades now has studied the sheep on St Kilda in great detail. And this is this fantastic island that's just off the west coast of Scotland. I say just, having never been there, because it's an awful long way to get there, and it's quite a difficult crossing. But these are some fantastic pictures that Iris took when she had a field season there this year that really capture just the ruggedness and the challengingness and the remoteness of the island where these sheep live. And these sheep have been studied for a long time on this island, and they really have provided a wonderful opportunity for us as people interested in vitamin D biology to understand more about this using this unique and very, very well-studied population. And some of the things that make it really attractive to us is the fact that they come in these 2 different skin coats has been one example. So I'll show some data in a minute that allows us to really start to understand more about the influence of food-derived vitamin D versus one vitamin D that's been derived from production in the skin. Because these animals are on this very tight island, there really is absolutely nowhere else they can go. It makes them incredibly useful for us to study, because unlike in humans where they go away on holiday and they just, might have a different diet for a few days before they actually have their vitamin D status, these sheep don't really have that luxury. They're very much restrained on this tiny island, so they have this restricted range and this real inability to then cheat the system and manipulate their vitamin D status. And because they've been so incredibly well studied, we know an awful lot about their history, how many lambs they produce, what happens to their lambs, how long the sheep survive, how they grow. All of this sort of thing is incredibly useful. And most importantly of all, these animals are blood sampled every year. So we're able to take these blood samples that have been meticulously archived for a number of years and then measure vitamin D within them. And so this really, the reason the ecologists and the geneticists are interested in this is because they're interested in what happens to the population. So this is just the year from 1985 through to pretty much the modern day here. And what we have is just the number of sheep on the island here. And this is it just fluctuating up and down. So the ecologists are very keen to try and understand, well, what drives these changes? What drives the population to suddenly surge and then suddenly crash? And that's what they've spent all their work and interest on. And what we've done in just these last few years is that we've measured vitamin D metabolites within these archived samples, and we've then started to understand much more about, well, what controls vitamin D status in these sheep, and then actually what does vitamin D correlate, or what does it associate with in terms of the life outcomes of these animals? And so the very first thing that we were able to show with this was really the relationship between coat colour and actually development of vitamin D status. And what we have here in the dark-coated sheep here on the left-hand side, we have much lower levels of vitamin D than on the right-hand side, the light-coated ones. But this is only the case for the vitamin D3. And this is just to sort of explain that in a bit more detail. The vitamin D that we talk of really comes in what we would call two flavours. We have a vitamin D3 that comes from foodstuffs, but it also comes mainly from the sunlight and the conversion within the skin. And then we have a D2 metabolite, where this pretty much only comes from plant-derived vitamin D sources. So what we can see here, there's an influence of coat colour on the vitamin D status in these sheep. And what we have is this very, very nice, what we would call in-house control, within sheep control. And what we see is that for the D2 that we know comes from the grasses, there's no different. So this really is very suggestive that the coat colour in these animals is really having an influence on their vitamin D status. And it's exactly as we predict. If you've got dark-coated skin, it's much harder for you to produce vitamin D, and so your vitamin D3 status is lower. And since we've started to understand more about the relationship between vitamin D and these sheep, the big thing that we've started to really get quite interested in is the relationship between vitamin D and what we would call the ecological fitness of the animals. And by that, we really mean, from an evolutionary point of view, the number of lambs that they then produce and the number of lambs that then go on to survive until they themselves reproduce. So what we've done over these series of years is that we've measured vitamin D in these sheep in late summer, and then we've then tracked them over the following year and seen how many lambs were born the following spring. And when we plot that, a number of lambs born, we've got 0, 1 and 2 here, and the vitamin D status again on this left-hand side, we see this stepwise increase in vitamin D status depending on how many lambs were being born. And so this has made us really interested and quite excited about this idea that potentially vitamin D status may be influencing their ability to then in essence, have increased reproductive success in this scenario. So again, we're very keen to say this isn't causation, but nonetheless, this relationship that is very, very striking, is consistent across all years that we've studied, is something we're now starting to spend a lot more time to understand. And this population has been really remarkably helpful and continues to be remarkably helpful for allowing us to understand much more about some of the really key issues around vitamin D biology that really influence all structures and all species, really. And one of the questions that we're very interested in is how stable is your vitamin D status? So if I was to measure your vitamin D status today, what would it be like this time next year? And so we've been able to do those sorts of studies, and what we've been able to show, and these are again great pictures that Iris took when she was away and painstakingly went through all the sheep that we were interested in tracking and got these great photos. So these photos live on the same tiny island, an island without trees, so very difficult to get away from sunlight or protect yourself from that. And really they're very, very similar in size and in appearance. And this one has this persistently low levels of vitamin D, whereas this one on the top here has these much higher levels of vitamin D. And so they're very, very much stable within years, but they also remain strikingly different. And so understanding the relationship and the reasons why this is something we're really interested in trying to understand. Is it the genes of these animals that control this, or is there some environmental exposure that we still don't fully understand? And this is something we're actively researching at the moment. And I couldn't really finish my section without highlighting the work that we're doing in the zoo with everyone's favourite animal, the penguins, which have been a wonderfully interesting project, because these are, again, have given us great insight and interest in trying to understand more about the relationship between vitamin D and health outcomes within the zoological species. But again, it really has allowed us to probe this issue, you know, how stable is vitamin D status within an animal species? And exactly as we've found in the sheep and exactly as we've found in the dog, it really sits at almost exactly the same level year on year. So understanding what causes that is something that we're really, really interested in. And really, everything that I've told you about so far is very much based on the clinics and the patients that we see within our hospital, and also some of the associative and epidemiological evidence that links vitamin D with health outcomes. But really, we have to take a very different approach, really, to try and understand, well, actually, is this causation? Can vitamin D really influence cells beyond the skeleton? And particularly, can vitamin D influence the immune response? And that's the point upon which I'm going to hand over to our scientist, and Iris, who's going to talk through some of the work that we've been doing in the lab together.
Thank you very much, Rich, for this great overview of how vitamin D does not only help keep our bones healthy, but how high, not too high, but high vitamin D levels are associated with a number of positive health outcomes. And as Rich mentioned, the important step is now to go from associations where we know that one thing is likely to coincide with the other to causation. So does vitamin D actively influence these health outcomes? And in order to do this, we have to use an experimental setting where everything else is controlled, but we can influence vitamin D levels. And ideally what we want to do is we want to understand whether vitamin D affects health on an organism level. Can it help prevent disease or even cure it? And then go down towards understanding what is it in our body that is affected by vitamin D, which cells are affected. And even then going one step further down and then figuring out which molecules are involved in this process. As Rich mentioned, our lab is particularly interested in how vitamin D modulates the immune system, and this particularly in the context of autoimmune diseases such as multiple sclerosis. Before I jump into experimental work, I thought I would give you a quick overview of the immune system. What is it and what does it do? What is its main function? If you think about our body, you're probably aware of the concept of it consisting of different systems, such as your cardiovascular system, your digestive system, or your nervous system. And these are quite easy to grasp because you can associate them with different organs that have specific functions, such as the heart or your gut or your brain. So what makes the immune system harder to grasp is that we can't really associate a specific organ and say this is where our immune system works. And this is because our immune system is spread throughout our body and consists actually of a multitude of cells that have each their specific functions, and they work together to fight infections and keep our body healthy. So to give you a bit of a flavor of the immune system, I'll start naming a couple of immune cells. Nobody needs to remember them. I'll tell you which ones to remember. So we have neutrophils, eosinophils, basophils, NKT cells, natural killer cells, mast cells, macrophages, and dendritic cells. And these are only a few. So you can already see that it's a highly specialized system. And as Rich mentioned, there are actually two arms to it. There's the innate immune system, which is what these cells belong to, which you can see a little bit like the foot soldiers of an army. So these are divisions that are skilled in seeing patterns in their environment and understanding when something is an infection. So if a bug comes along, these guys are able to recognize it and fight straight away. But within those, there are also cells which are really good in reporting back to headquarters, basically. So dendritic cells here are really good at understanding there is something wrong and then going to meeting rooms where then talk to the so-called adaptive immune system, the other arm of the immune system, to fight infection together. And so part of the adaptive immune system are cells called B and T cells. For our talk, you only have to remember dendritic cells and T cells. But the uniqueness of the adaptive immune system, actually there are two, are that the adaptive immune system is able to refine weaponry. So if you think of the foot soldiers just recognizing something and fighting, the T and B cells take the time to reconsider, to basically refine the weapons to fit perfectly with exactly that in that bug that you've been seeing, and therefore enhance the chances of you being able to eliminate it. And the second unique point of the adaptive immune system is that it creates memory. So once an immune response is triggered through the communication of dendritic cells and T cells, and thus the weapons have been refined, some of these will be stored, which means that later on in life, if you see exactly the same bug again, your immune system is ready to attack and eliminate the invader before you even realize it. And this is, by the way, how vaccinations work. So the principle is exactly this one. You give either bits of a bug or a mild version of a bug which cannot induce disease, and this triggers this response, which means that afterwards you have a long-lasting memory, and if the actual disease comes along, you're ready to tackle it. So where is all this happening in our body? As I said, the immune cells are spread throughout our body, but they're so-called lymph nodes, which are the meeting rooms for these cells. And in here, they are the green dots that you can see all over our body, underneath our jaw, in the maxillary region, or in the groin. And the ones underneath your jaw are probably the ones that you all are aware of, which are the glands. And you probably have experienced this yourself. When you're ill, they can be physically swollen. And this is actually when your dendritic cells and T cells have a very heated discussion about what to do next. And here on the right, you can see that, let's see if that works. Oh. Pointer. Nope. There we go. So you can see that these cells move. And so you can see the T cells in green here, and they'll be moving around trying to see if they can find a dendritic cell, one of the reporter cells, to communicate with. And once these dendritic cells come in, they will have an interaction. To make a bit more sense of this, here's a cartoon. So our dendritic cells, which are part of the innate immune system, will be walking around our body, sampling tissue, and trying to see whether anything is wrong. If we do have an infection, and the dendritic cell samples this, it will be able to recognize it's an infection and becomes activated and angry. And it will take the cargo to the nearest lymph node and try and find a T cell that matches this bit of the bug. So Rich was talking about before about receptors. So receptors are basically molecules on the surface of cells that are able to recognize a certain bit of a bug. And each T cell in the lymph node will have a different receptor. And so the dendritic cell will need to find the right matching T cell, which, like a lock key system, will fit perfectly. And only the T cell which recognizes this bit of the bug will be activated. And being activated means, in the context of T cells, that they'll refine their receptor to fit even better. They'll proliferate, so they multiply, and they'll go and search for the tissue that is infected and help fight infection. And in the case of our body, the immune system is an amazingly good system, in the sense of, if you think about all the food that you eat, the different types of food, all the environmental things that you come in touch with, in contact with, or even more the microbiota, which you might have heard of, the bacteria that are actually helpful, that are in our gut or in our skin. If you think of all these different influences, how our body, our immune system, is able to understand what is to be tolerated or even protected, such as our own cells, and what is to be attacked because it's harmful. Because how would it make the difference between microbiota and the infections that would be harmful to us? Or even in context of a cancer, our immune system is very good in eliminating damaged cells that might lead to tumor formation. But in essence, these are our own cells, but they have something different. They have a mutation, and our immune system is able to see this in most cases. But as with every system, it's never flawless. And basically what can happen if you have a system where your immune system is overexcited, it might see parts of your own body and think that this is harmful, and therefore mount exactly the same immune response as before, where we'll take samples and accidentally get activated, and this dendritic cell will then go, and if it finds a matching T cell, this T cell will get activated and try and fight this tissue that is actually our own body. And this is what can lead to autoimmune disease. So and what we are really interested in is whether we can understand how vitamin D can modulate the immune system, and whether it helps keeping that balance between what to tolerate and what to fight against. This brings me back to the map that Rich has shown previously, where we've seen that in the northern states, the prevalence of multiple sclerosis, which is an autoimmune disease whereby T cells attack parts of the brain that are myelin, which are the sheet that protect our neurons, so the prevalence is higher in the north and less so in the southern part, where there's more sunshine. And the question being whether vitamin D, which we can make from sunshine, is involved in this. So to bring this back, though, to where we are, I just wanted to have a quick show of hands and ask whether how many people you believe are affected by multiple sclerosis in Scotland. So we have three options, either 1 in 500, 1 in 2,000, or 1 in 5,000. So who thinks that 1 in 500 people in Scotland are affected by multiple sclerosis? Okay, approximately a third. Maybe it'll continue like this. How about 1 in 2,000? That's half, probably. Okay. And 1 in 5,000. Oh, my math don't add up. So almost none. Unfortunately, it's 1 in 500. 1 in 500 people are affected by multiple sclerosis in Scotland. And if you look at the world map, as Rich said, there is a bit more confounding factor in the world map compared to one single country. But the most important thing is that we are actually leading the world with the incidence of multiple sclerosis. So it is a matter that is important to us, and trying to understand how we could potentially manipulate the risk of disease is really important. So to do this, as I said, we need to have an experimental approach. And to understand how vitamin D can modulate a complex disease like multiple sclerosis, we need to use an animal model where we can control the environment, but then manipulate the vitamin D levels. So obviously, animal research at the University of Edinburgh, as well as in the rest of the UK, is highly restricted in that we have to justify the use of animals for our research. And the first question being, is it necessary in order to answer our question? And really the concept that we all try and follow is the concept of the 3 R's: replacement, reduction, and refinement. So first we ask, can we use a non-animal model in order to answer our question? And if we need to use it, can we reduce the number of animals? And again, when we then do our animal research, can we refine the experimental settings to ensure animal welfare? So in our case, we have 2 models of multiple sclerosis, which are mouse models, which basically mimic the symptoms, or better, the clinical signs of multiple sclerosis. So you might have to bear with me while I go through the models with you, because they're really important in understanding why, which part of the immune system is being affected by vitamin D. So what we do is we take T cells that are reactive against myelin, which was the bit in the brain which is being attacked by T cells in multiple sclerosis, and then we put them into a mouse and immunize the mouse with myelin components, which means that what happens then is, as I showed previously in the cartoon, is that dendritic cells get activated and present these bits of myelin to the T cells, which then get activated and go to the brain and damage by attacking the myelin. And on the right-hand side, what you can see is how we assess the clinical score of these animals, whereby signs that are similar to what a multiple sclerosis patient would have in a relapse are monitored, and they go away by themselves, so it really is like a relapse in multiple sclerosis. Our other system that we use is kind of a more passive disease model, as we call it, because what we do is we jump over this communication process and we pre-activate these T cells outside the mouse and then put them in, which means that it's just these activated T cells that will go straight into the brain and cause the damage. So what we did is we used these 2 models, and we gave half of the group no treatment and half of the group we treated with vitamin D during the initial stages of our disease induction. And so what happened here, as you can see very clearly, is that when you give vitamin D at the beginning, and here in red, these mice within the same cohort, they don't develop this multiple sclerosis-like disease, whereas the control ones did. So clearly vitamin D is able, in this model, to prevent disease. However, interestingly, what we found when we used the passive disease so where this communication isn't going on, that the vitamin D group was no better off than the control group. And this really means that vitamin D seems to be acting at the beginning, so where the dendritic cells are communicating with the T cells. And this is really important to understand if we think of how we portray vitamin D as a healthcare cure. If this was to translate, that it's really important to understand that the beginning of our lives and before the induction of disease, vitamin D might be important, but once the disease is established, it might not be very helpful anymore. So, on an organism level, we've now shown that in a model of multiple sclerosis, vitamin D is very useful in modulating and dampening the immune response. And we decided that, dendritic cells, who are the reporter cells communicating, might be the target of vitamin D. And so we wanted to understand more what vitamin D does with these dendritic cells. So what we do in the lab is we culture dendritic cells. We can do this in a dish like this one. It's sterilely packed, so we do this in a hood, not here. Now it's not sterile anymore. We can then culture progenitor cells, so precursor cells, in these dishes to make them into dendritic cells and ask questions. In vitro is what we call it, so not in an animal. But what we can do is, as in an animal, we can either leave them or we can activate them and make them angry by giving bacterial components. And what we did was do the same in the presence of vitamin D, and then ask the very general question of what changes, to see whether we can pick out any changes between these groups, any difference that vitamin D makes that might account for the effects that we see on an organism level. And just to walk you through this, we looked at, and we can have huge assays where we can look at lots of genes. So we had 20,000 genes analyzed, of which hundreds of gene products were altered. So there were differences in levels between those cells. And in the end, when we looked at comparing just the vitamin D groups with the no vitamin D groups, there were 6 gene products enriched. And as you do, at some point you have to make choices in life. So we went for one particular product, and this is called CD31. So we realized that vitamin D was making dendritic cells have highly enriched levels of this molecule called CD31. Now, not much is known about this molecule. It is known to be present in our vascular beds and to direct white blood cells into certain parts of the body. And it is known that it is expressed, so basically it is present on several immune cells. But what it does in dendritic cells is unknown. So now we are going, if you think about our levels, we had the organism level where we understood that vitamin D was affecting health. Then we went and looked, found that it was dendritic cells that it was affecting. And now we want to know, how does vitamin D affect dendritic cells on a molecular level? And so this is where CD31 comes in. And we try to understand whether CD31 was important in inhibiting immune activation. In order to do this, we use a little trick in the lab where we asked cells to express, so basically to make the CD31. So nothing is different between those two cells apart from the fact that we've now infused the levels of CD31 in one of them. And then we asked the question whether just by altering the levels of CD31, this would affect how these dendritic cells can activate T cells. And what you can see here is we have a measure of immune activation on the left-hand side on the y-axis. And what we do is we put the same amount of T cells together with increasing numbers of dendritic cells. And you would expect that with an increase in the number of those activating dendritic cells, the overall immune activation would become higher. And you can see in your control group here, in black, that this is indeed the case. So the more dendritic cells you put together with the T cells, the higher the immune activation becomes. But what you can also see is that the CD31 infused group is lagging behind. So at the same number of dendritic cells put together with the T cells, your CD31 infused group has a lot lower immune activation than your control group. So clearly CD31 itself as a molecule is able to dampen the immune activation. And then you can take it one step further. So that's what we do. We just ask the next question. We wanted to know, how does CD31 do this? How does CD31 influence immune activation? And we had this hypothesis that if you think about your dendritic cell and your T cell and their interaction defining immune activation, that potentially the interaction time would be important. So if you think about a communication, if you have the time to explain what's going on, somebody needs to be invading, we need to do this, then that will lead to activation and to a good signal. Whereas if you only have a short time of interaction, you won't be able to get the message across. And in order to look at this, what we did is, again, made these dendritic cells either with nothing or with infused CD31, and put them together with T cells and took images every 30 seconds to make a time-lapse video. So we could then measure how long were these cells talking to each other. And this then looks a bit like this. So here's your dendritic cell. This is a bigger blob. And then the smaller blobs are T cells that are walking around. And you can see that this T cell, for example, is interacting with the dendritic cell for a while. It's still there, thinking. And then it decides to go away. So you can then count the times of frames, the amount of frames that you got, and therefore the interaction time between the two. Now, when we count this up between the two groups, what happens is that we can see in the control group about 30% of the interactions that we counted were less than 5 minutes long. Now, if you look at the CD31 infused group, this has doubled, and the amount of interactions that last for a shorter time than 5 minutes is about 60%. So this confirmed our hypothesis that this molecule, which is enriched through vitamin D on dendritic cells, called CD31, was shortening the interaction time and thereby leading to a reduced activation of the immune system. So just to sum up on this experimental part, if I've lost you in between, what we think is happening is when we generate dendritic cells in the presence of vitamin D, this leads to increased levels of a molecule called CD31, which in turn means that when they try to activate T cells, which will be the ones delivering the immune response, the interaction time is down and therefore the activation is reduced. Now, there are lots of more questions coming up, obviously, at this stage, and this is something we are addressing in our lab. And we can obviously understand that CD31 is involved in this process, but there will be lots more that vitamin D is doing. So bringing this back up to the level of how we know an immune response is mounted, how do we think vitamin D fits in here? What does vitamin D do? So when dendritic cells accidentally sample healthy tissue and believe that they need to attack it, which could lead to autoimmune disease, the presence of vitamin D might be able to moderate this activation, which then leads to reduced T cell activation and no damage. So what I've hopefully been able to show you here is that following on from what Rich's work is doing by looking at different types of animals and associations between vitamin D status and health outcomes, what we do in the lab is to tease apart the mechanism behind this and how if vitamin D actively is involved in these processes. So we've taken away that question mark. In this case, there's lots of different things that vitamin D might be doing, but what we've looked at is a mouse model of multiple sclerosis where vitamin D was able to suppress the induction of disease. And we found out that dendritic cells are involved and affected by vitamin D. And at least partially, this was able to be was explained by the elevated levels of CD31 on these cells. So this is kind of just to give you a flavor of how we go about understanding vitamin D biology in the lab. And this brings me back to the overarching goal of how we try and integrate different aspects of vitamin D biology and addressing this from different angles. So using patient samples and patient history to understand different diseases or illnesses that can arise that have to do with vitamin D. Using population approaches where we have wild populations and we can ask big questions where you need a big sample size, and then taking hypotheses that arise from these into the lab and trying to understand the mechanisms behind vitamin E biology. And with this, we would like to acknowledge everybody involved, which includes funders and the shooting stars, the animals, and also to point you out to our website that our lab has, where you can have a look at the research we do. We also have links to the publications and also the latest news on our public engagement. And if you wanted to get in touch, we'd be very happy to hear from you. And with this, I'd like to invite any questions that you have from the audience. Thank you very much.